I'm not really one of those people who work on CRISPR technology per se. We're really are users of this technology in the hope to employ this technology to generate useful mouse models for our functional study. So we live in the era of technology explosion, right? So I remember when I was a graduate student, if you can make a one knockout a model for a gene, then that's a beautiful graduate thesis. Usually it takes 3 to 4 years to make the knockout, and then people spend a year or two to characterize the phenotype. But now we're living in a completely different era. So I'd like to give you guys some of my perspective on witness the advance of the CRISPR technology and its impact on mouse genetics. Can I get a sense how many of you are working on mice, using mice as a model system? Okay, all right. So you guys have to bear with me a little bit for the intro for the mouse as a model organism. So mouse is one of the most powerful mammalian model organisms. It's similar to human. About 95% of the mouse genes are homologous to human. And mouse can be easily genetically manipulated, so you can make a whole bunch of really sophisticated genomic engineering in mice. You can generate deletions, you can generate insertions, you can even generate some of the genomic structure variants, such as chromosome translocation, chromosome inversion, chromosome duplication. So historically, these can all be doable, but it's pretty complicated. Mice has been bred for decades, and there are many strains of so-called isogenic or congenic mouse strains, meaning that these mouse strains, they are homozygous on almost every single locus. And this is a very important feature of the mouse genetics field, because that makes comparison of mouse phenotype much easier among different labs. Mouse have accelerated lifespan. In the laboratory setting, mouse lifespan is 2 years. So that's almost 1 mouse year is equal to 30 human years. So we are looking at accelerated progress in both development and physiology. And that enables mouse to be a very powerful system to study some of the late onset disease, such as aging, neurodegeneration, cancer in human. Also, mouse has been a model organism for almost a century, and there are many well-characterized biological systems to probe biological readout in the cell level, in the tissue level, in the organism level. So that gave us a powerful tool to probe human development and physiology. Mouse are very small. You can actually house them very effectively in the facility, and it's a very efficient in gestation. So they're a very effective research tool. So probably among everything that I said, the most important feature of mouse as a model organism is this ease to genetically manipulate mouse. Actually, for many years in the mammalian biology research, mouse was the only organism that we can easily manipulate the genome. And this is largely due to a group of pioneers in the field, which I'm going to tell you a little bit about history so that you can appreciate how amazing CRISPR technology enables us to do. So first of all, let me tell you about mouse development. So mouse development, there are 2 stages of mouse development: embryonic stage, where it happens in utero, and postnatal development. So embryonic statement can also be divided into 2 stages. So-called pre-implantation development happens from one cell embryo to a blastocyst embryo, which is about 100 or so cells. And so when the fertilized zygote travels through the oviduct, they continue to have asynchronized cell division. This is very different from, say, frog, where cells are dividing synchronously. But in mouse pre-implantation development, cells are dividing asynchronously. And then by day 3.5 to 4, they reach a stage called blastocyst, where the first cell phase specification occurs. So in this stage, the development can be completely recapitulated in vitro. This is a foundation of all the in vitro fertilization technique that later developed in human as well. So the fact that these embryo culture can happen ex vivo enable us to have an opportunity, a window of opportunity to manipulate the mouse genome during early development. And at day 4.5, the mouse embryo will implant into the uteri of the mother. So the first person that really pioneered the series of the very first step milestone events for manipulating mouse genome is Anne McLaurin. She actually was able to develop a technology where you can transfer pre-implantation mouse embryo to the uteri of a recipient mother. So this is very important. So that allows you to segregate the pre-implantation, the embryonic development into two stages. One stage in vitro, where you can manipulate, the other stage in vivo. So the next major advance in mouse genetics is the ability to be able to manipulate mouse embryo. Early on, people realized that you can do just a lot of amazing things about mouse embryo. You can actually divide them in half and let them develop into individual embryos at the 2-cell stage. You can actually also aggregate multiple embryos together, usually two. You can put two embryos together and make them into an aggregate. So this technology called Marula aggregation, where you take two different mouse Marula embryo. This is around day 2.5 of development. And then you can actually push them together in vitro, and then they will aggregate into a chimeric blastocyst, which can be transferred into utero by the technology that Anne McLaurin developed. And another really major milestone event is developed by Richard Gardner as well as Ralph Brinster. So they developed a technology to inject cells into the blastocyst stage. The blastocyst has a cavity, and then you can actually inject cells. In their case, they injected embryonic carcinoma cells into the blastocyst stage, and the injected donor cells actually end up integrated into the blastocyst, and then they contribute to certain developmental lineages in the adult animal. So the series experiment enable us to really have a donor population that we can manipulate, and then transfer the manipulated population into a recipient mouse embryo, and then generate so-called chimeric animal. But the most important invention, one of the most important technology events is actually the derivation of embryonic stem cell, because all these make it possible for you to generate chimeric, so that you can have a donor population that susceptible for manipulation. But what do you manipulate? So actually, Ivan Martin, as well as Gail Martin, in the late 70s and 80s, have derived a specific cell line called embryonic stem cells, and these are pluripotent stem cells that can give rise to all lineages in embryo, including the germline. So let me just quickly tell you about embryonic. I know Dirk has touched upon this. I'm not going to spend too much time. But basically, the one-cell zygote are totipotent. They gave rise to not only the embryo, but also the actual embryonic tissue, such as placenta and yolk sac. But by blastocyst stage, the first cell phase specification events is complete in mouse, giving rise to the outer large flattened trophectoderm cells, which eventually will develop into placenta, as well as so-called inner cell mass. And these are smaller aggregate cells that aggregate into one pole of the blastocyst, and inner cell mass will further differentiate into primitive endoderm cells and epiblast. So primitive endoderm cells will eventually develop into yolk sac, and epiblast will give rise to embryo. So you can actually derive embryonic stem cells from the inner cell mass of a blastocyst, and the embryonic stem cells have the developmental potential of epiblast cells. So these cells, when you inject into a recipient blastocyst, have the potential to contribute to all cell lineages within the embryo, including the germline. So having the cell culture in vitro is one of the most critical events to enable mouse genome editing. And actually, for decades, mouse is the only organisms where people can derive embryonic stem cells. Even rat is very similar to mouse, but it takes actually more than a decade before people were able to develop rat embryonic stem cell. So this window of time where people can manipulate mouse genome and generate a lot of interesting mutations and engineerings that enable mouse to be one of the most popular mammalian organisms. But I want to tell you, for certain diseases, mouse is not the best organism from the disease standpoint. Pigs, dogs, and rat, they all have their own advantages in modeling certain aspects of human development and physiology. All right, so now you have the cell line. You hope to be able to manipulate them. So the hero in this area is actually Mario Capacci and Oliver Smithis, and then they are together with Evan Martin was awarded Nobel Prize for their contribution to make mouse knockout a possibility. So they actually developed homologous recombination strategy to engineer the ESL genome. And then they actually reported the first successful gene targeting in ESLs in '89. And then the following year, we have seen the making of the first knockout mice in vivo. Because basically, all the pieces are together. You engineer the ESLs, and then you inject them into blastocysts and you transfer into utera. So, so this is the way how it's done. So showing here is a blastocyst where you have a holding peptide that linked to a vacuum. So by vacuum, you can hold this embryo very nicely into the holding peptide. And then people usually place the inner cell mass facing the side of the holding pipette. And then over here, you have an injection pipette where usually you can inject 10 to 15 ES cells, you can see over here, into the cavity, into the blastocyst cavity of the blastocyst. And then after you finish the injection, this transferred to mouse, a recipient mouse, and usually the donor and the recipient are distinguished by the different co-color genes that they carry. So the recipient can be albino in this case, and then the donor ESL carry a dark co-color gene. Therefore, you can actually see the mosaicism by the extent of the co-color mosaicism for the founder mice. And these mice will be further bred to wild-type mice to generate heterozygous mice. Another strategy, equally efficient but less popular due to the extensive, labor-intensive procedure, is aggregation. So you can actually have two mouse A-cell embryo, and then you use so-called sandwich method. So this is a clump of ES cells. So you make a little dent in your plate, put these two embryo, sandwich this embryonic stem cell cluster, and then you culture them overnight. If successful, then they will become one single chimeric blastocyst, and that can be transferred into a recipient pseudopregnant mother. All right, so then another really important thing, which are less talked about in the CRISPR era, but it's talked about a lot in the ESL era, it's the germline transmission. So sometimes you have ESL contributing to all somatic lineages, but they fail to give rise to germline. So having a good ESL is not trivial. So this is actually chimeric mice, which are actually very much dominated by the donor population. So the progeny of these mice are all pigmented. So you know that the germline, the ESL derivative undergo germline transmission. All right, so one last thing I want to say about the history is the tetraploid complementation. This is the ultimate way to generate ESL-derived mice. So the idea is that you can actually aggregate a tetraploid embryo with a diploid ES cells. The tetraploid lineages can give rise to actual embryonic tissue. Actually, many of the cells in placenta are tetraploid or aneuploid. And but only the diploid ES cells can give rise to embryos. So by doing so, you will be able to generate a mouse that are 100% derived from the ES cells. So this is actually a technology developed by Andrea Nagi's group in Canada, and has been very successfully used to enhance the efficiency of gene targeting. So this is just to summarize what you do for making a knockout these days using ESL technology. First of all, you have to design a targeting vector to facilitate homologous recombination. This can take anywhere from a few months to half a year, and then you have to screen for the clones. And then after, you have to screen for the successfully edited ESL clone. And then after that, you will be able to perform blastocyst injection, usually through a transgenic facility, and then get germline viable, generate viable fertile chimera, and then that undergo germline transmission. So you have to cross another generation to expand your colony and get true heterozygous mice. So this whole process, if you're lucky, takes 9 to 12 months. Another parallel technology in the mouse genome editing is the transgenic mice, and this is a lot simpler procedure-wise. So basically, what you do is you make your transgene, usually a gene that allows you to overexpress a particular protein, so you basically inject into the so-called pronucleus of the zygote. So pronucleus is the nuclei of the gamete before they undergo nuclear fusion. So between fertilization, the initialization of the fertilization, until the fusion of the two nuclei, there's a time window where you can perform this injection. So you can clearly see there are two visible pronuclei in this zygote. After the injection of the DNA molecule, you can see the expansion of the pronuclei, and this is how a successful injection should look like. And I want to tell you that this is nothing trivial. As a graduate student, I tried this technique for like a month, and I finally have to give up, and realizing that I really need to spend 3 to 6 months just perfecting this technology, because hitting the pronuclei is nothing trivial. But in this case, it's fast because you don't have to do a lot of manipulation for your targeting vector. Germline transmission is often very easy because you're manipulating the embryo, and then usually you're not dealing with extended in vitro culture. And then also, but the challenge is that there's a limiting editing capacity. You can't really do much by integrating a DNA fragment into the mouse genome. It requires pronucleus injection. But you could maybe see the phenotype in the founder generation. So there are pros and cons in this strategy. So I listed the comparison over here. So what I want to emphasize is what CRISPR really enables us to do is marry the advantages of both technology and then create a technology that's really easy for people in the mouse community to use as a tool to probe genetics. So there are two major type of CRISPR editing in mouse. Probably the most talked about is the germline mouse editing, in which the CRISPR system were delivered into a zygote, and this lead to a transmissible genetic alleles. And then, so the current technology actually enable multiplex genomic editing, so you can target multiple genes. There are reports about 5 genes targeted simultaneously. And then also the designs are much easier than the ESL targeting vector. The manipulation is also a lot easier, which I'm going to come to in a moment. So the general sentiment in the CRISPR field for mouse editing are this. So there's a strategy where you can do so-called one-step CRISPR editing, in which you deliver all the CRISPR component into the mouse zygote. And this strategy is really fast, it's efficient, reasonably efficient, and has generated a lot of very useful mouse models in proof of principle experiment. But this is only applied for simple editing. for very complex editing, people still want to find the comfort zone of ESL system. They oftentimes either use homologous recombination or use CRISPR editing on ESLs. This enables them to really precisely genotype what the editing events truly are before they move on to the mouse. So basically, I'm going to cover these two different approaches to targeting for different targeting schemes. In addition to germline mouse models, CRISPR technology has also been widely used for somatic mouse modelings. And somatic mouse modeling has actually been quite popular. This is largely due to the efforts on two fronts. One front is the delivery methodology to a variety of somatic tissue in mouse. And this enables us to deliver all kinds of genetic components to specific organs or specific tissue type. And this has been in existence for decades by now. And liver is perhaps one of the most powerful organ for somatic delivery of genes. And many of the clinical trials actually are derived from some of these early studies to pursue gene delivery in liver in vivo using mouse as a model. The other front that has been very powerful is decades of research on mouse somatic stem cells. In this case, these mouse somatic stem cells could be perfect cells for targeting, because if you were able to manipulate somatic stem cells in mouse tissues, their progeny will carry the desirable CRISPR editing events, and then that will enable you to study CRISPR phenotype in a tissue-specific manner, also in a controllable manner. So this kind of editing scheme actually enables tissue-specific and inducible CRISPR editing, which can be very powerful to study certain aspects of developmental process as well as physiological processes. So there are two major approaches to get to this tissue somatic mouse modeling using CRISPR. One is to achieve tissue-specific delivery of the CRISPR system, which I'm going to talk about in more detail in a minute. And then the other approach is to generate inducible Cas9 mouse models, which will enable somatic editing in specific tissue type under specific time frame. So I'm going to also cover these efforts for the mouse community. Okay, so I talked about the two different types of editing. What can CRISPR do in mice? So we can certainly make gene knockouts, and people have been making gene knockouts for decades. And we can do not just doing indel, but we can also do some simple modifications, such as small insertions, small deletions, point mutations. So these kind of gene knockouts and simple modification are best achieved by those one-step CRISPR editing in mouse. I think that there is a consensus that has been reached in the mouse community that this is very doable, it's highly efficient, although it really depends on the design of the guideline in particular cases, but in general, this is pretty much replacing the ESL technology for genome editing for mice. There's another type of editing which is involve making very complex genomic structural variations in mice. And this involves, for example, making very large deletions, large duplications, making reciprocal chromosome translocation, or making large chromosome inversion. And then for these kind of very sophisticated editing, historically, people try to engineer different LOXP sites in different chromosome regions and then induce these chromosome rearrangement by Cre-mediated recombination. But those are very, very time-consuming. It's very difficult for average lab to achieve. But nowadays, people can achieve that using CRISPR editing. The challenge is that you usually have very complex editing events. To be able to select the ideal, the desirable editing events, the best thing to do is actually to do this in ESL, and then you derive individual ESL clone, and each clone contains one single edited events, and then you can do a southern to really validate these edited events before you go into mouse. Otherwise, you will end up with a pool of complex genetic events that's hard to sort out in vivo. So still the sentiment is try to achieve this in vitro mostly, but there is one successful story which I'm going to tell you that people were able to achieve this in vivo, and then they achieve this because they have a selective pressure to amplify these cells in vivo. All right, so let's talk about this. So the first effort to look at mouse editing is to use CRISPR genome editing in mouse ES cells. And not surprisingly, this work was first published by Rudy Janish's lab, who are always at the frontier for genome editing for mouse, starting from zinc finger days, Thailand days, and now CRISPR. So the way that they want to do this experiment is they take a family of genes called TED. These are a family of enzymes that mediate the first step of DNA demethylation. So they convert 5' methyl cytosine to 5' hydroxyl cytosine. So this is the first step of DNA demethylation. These enzymes have overlapping but distinct expression pattern. They regulating a large number of early developmental events. So people have knocked them out individually, but knocking them all out altogether has been a challenge. So what Rudy's lab has reported is they were able to target ES cells for all 3 genes simultaneously, and shown here is their R flip results as well as southern results, demonstrating that they can acquire ESL clone with simultaneous biallelic targeting on all 3 genes. So they reported 20 out of 96 in their success rate for biallelic editing on all 3 genes, and that was extremely high. They even went further and then to target 5 genes altogether, and 2 of which are Y-linked, so you only need to target 1 copy. The other 3 are TED1 through TED3, and they were also able to report a very reasonable success rate. So this is what's happened in vitro. In vivo was actually very interesting. The very first paper that report CRISPR editing didn't come from any of the major mouse labs. It come from a very small lab, and this paper published in a less prestigious journal. It was fast study and not so very well documented. It's very rushed, but sometimes it's actually interesting to see small lab that were able to make things like that happen. So this is a study that done not designed perfectly, but actually illustrate all the principal technology that we're using now. So basically, what they are doing is they take Cas9 mRNA and then in vitro transcribed guide RNA, and then they design a guide RNA against GIP, and then they're targeting GIP in the transgenic mice, which is expressing ALK4 ARIS GIP. So these mice can actually express GIP under the ALK4 promoter. And so they did zygotic injection. And what's really amazing is they reported you don't need to do pronuclear injection. I told you how difficult it is to do pronuclear injection. So here, they only need to do cytoplasmic injection. And a lot of people can do that if you remove the requirement for pronuclear injection. So then they reported 5 animals, and then 1 out of 5 was edited by NIGJ. So that was actually the very first paper in the mouse community for the CRISPR genome editing. And 4 months later, the more comprehensive study came from Rudy Janish's lab. They pretty much use the same strategy. They take Cas9 mRNA, and then they use multiple guide RNA targeting multiple genes. They did zygote injection into cytoplasm. This is so much easier. And then they transfer them into recipient mice. And then they were able to recover mice that are correctly edited. So in the early days, a major consideration for the mouse community when it comes to a new technology for genome editing are the following. So first of all, we really don't know how you should deliver Cas9. Should you deliver mRNA or DNA? More recently, people talk about RNP. So this is a issue that needs to be resolved. How about editing efficiency? Is it comparable to mouse ESL editing? So this was a question in the field. Is Cas9 toxic to the mouse embryo? We have absolutely no knowledge. What about germline transmission? Is Cas9 delivery going to impact the efficiency of germline transmission for this procedure? And finally, off-target effects. Is off-target effects going to affect our experiment? So basically, in the very first paper, they just reported using Cas9 mRNA plus guide RNA for zygote injection, and then they reported the live birth rate is 10 to 20%, meaning that if you transfer 100 injected embryo, you will recover 10 to 20 mice from the progeny. So this may seem very low to many of you who don't work with mice, but this is very standard for mice. So that means that Cas9 has very low toxicity. And then the efficiency of NHEJ editing was astonishing to many of us in the field when the paper first came out. So as you can see, this is a targeting efficiency for individual genes. This is actually using different concentration of Cas9 mRNA and guide RNA, so you can see the efficiency is very, very high. And just to let you know that in general, if we can get about 20–30% of editing efficiency through ESL type of technology, where in general people are very happy. So this is very, very high. And then they can also do multiplex targeting for type 1 and type 2 simultaneously. And then they reported about 22 out of 28 was successfully targeted on 4 alleles. So this is a very amazing efficiency. And then they went on to do HDR editing because people really want to have a designer way to edit mouse genome. So they did a pseudo HDR experiment, in my opinion. Well, many people do that. In which they find a restriction enzyme that close to a PAM site, and then they engineer a donor, a single-stranded DNA donor that replaces the restriction enzyme by EcoR1. And then using this strategy, they were able to achieve about 20% biallelically edited mouse with HDR editing. I want to emphasize that this type of HDR efficiency, you need to be a little bit careful, because when you really want to design designer editing, and you are so limited by the availability of PAM sites or restriction sites that you can do easy targeting. So this may not be so transferable to your own experiment. But this can be done. So on a single gene level, they reported about 90% HDR efficiency, so which is very high. Many people may not be able to recapitulate this level of success, but in general, this type of strategy is actually as efficient, if not more, than the ESL technology. So people are very happy about this technology. And then you can also insert a small fragment. So this is another paper came out about 5 months later from Rudi Janish's lab again, and then in which they actually insert a 42 nucleotide sequence into the SOX2 genes. And then this is a 42 base pair nucleotide, 42 nucleotide sequences actually encode a V5 tag, which enable you to do immunofluorescence staining to detect the endogenous protein. Again, this is a proof of principle experiment. They reported about 30% efficiency. Again, this is very high. Traditionally, this type of experiment will have much less efficiency. And then another really exciting finding that they report is they were able to insert a large fragment. So in this case, they try to insert a mCherry reporter and fuse to the endogenous NANOC gene. So they actually injected a plasmid donor together with the Cas9 mRNA and guide RNA, and then the plastic donor contains 2 kb and 3 kb homologous arm on each side of the targeting vector, and then they have the mCherry reporter as an inserted sequence, and this also have been reported with 10 to 20% efficiency, editing efficiency. So the take-home message is, if you want to insert a larger sequence, you have to use a circular DNA donor. If you only insert something that's short, such as a tag, you can use single-stranded DNA donor together with Cas9 mRNA and guide RNA. And the most important thing, one of the most important sequence that people want to insert it into the mouse genome is a LOXP site, which is about 35, 36 nucleotide in length. So the ideal case is you want to make conditional knockout, right? So then you basically hope that you will be able to flank a important genomic fragment by 2 LOXP sites. So Rudy Janish's lab also tackled this very important challenge for many people, which is cared by many people in mouse community. So they are looking at a very important gene called MECP2. This is a gene that ubiquitously expressed in all neurons, and mutation of MECP2 causes Rett syndrome. So they want to make a conditional knockout of MECP2. So they design 2 donor DNA oligo flanking the exon 3 of MECP2. And then they actually inject 2 guide RNA, 2 different DNA donor, as well as mRNA for Cas9 into the zygote. And then again, you can envision that there are many different ways that this integration can happen, right? So you can have 2 LOXP sites inserted into 2 different alleles, where you can have 2 LOXP sites integrated into 1 allele. The only one that's useful is when 2 LOXP sites integrated into 1 chromosome, and that is about 20% of the efficiency that was reported. However, this technology, this strategy has a fundamental issue when transferred to other scenario, because oftentimes when you do this, you could get large deletion rather than the desirable flux allele. So the current days, people are tend to use plasmid donor as a way to engineer this flux allele of a particular gene, rather than using 2 separate guide RNA and then 2 separate single-stranded DNA donor. So there are still debates on whether this technology can be, whether this strategy can be further improved to enable a simpler design for this approach. Yeah.
It's very interesting efficiency of 2 different HDR at 1 locus, and that has to be way higher than the product of independent probabilities. So does that mean that if a locus is primed for HDR at 1 spot, it much more efficiently incorporates HDR at an independent event?
So I have to say that we tried this experiment. What we see most happen is a deletion. So when you do design this 2 guide that cut different region of the mouse genome on the same chromosome, if they're not too far apart, and you usually get deletion rather than these type of integration. I mean, it is possible that their particular guide and their particular design enable that particular experiment to get very high efficiency, but when other people try in different system, it is not that easy. So, and also people do emphasize that when you have this type of design, you want to test your guide RNA so that at least their cleavage efficiency are comparable. You certainly don't want to have a very, very efficient guide and a very inefficient guide as a pair, because then you could also generate an IGI type of editing in the very efficient guide cleavage. So, and another word of warning is people have told me that you absolutely have to do the southern genotyping as well as sequencing to conform, because you may not getting what you think you get. So, Okay, so advantages of this one-step CRISPR editing in mice is simple design, no need for complex cloning for targeting vector, cytoplasmic injection into zygotes, no need for polynucleus injection. There's low toxicity, high rate of germline transmission. Actually, as a matter of fact, I've never heard people complain about germline transmission, which is great. And then multiplex editing is possible. So, okay. So for the delivery method for CRISPR editing in germline models, it's actually quite interesting. So people have tested this extensively. The very first study to compare mRNA and guide RNA injection versus linearized DNA injection, and clearly mRNA and guide RNA injection achieve much better editing efficiency. And then another study published by Jin Sung Kim's lab in about a year later, and then they show that if you do Cas9 RNP injection, and you can get a further improvement of editing. So in their report, they achieve about 90% NHEJ editing. So now, after all this implication of the CRISPR technology, the key challenge now, if you think about the procedure, you need to inject those, transfer them into pseudopregnant mother, and then recover founder mice, genotype them, and mate. The key challenge now become microinjection, which is something that people take for granted. You just give it to your facility to do it. But now the most time-consuming bottleneck is microinjection. So there's a number of groups that are looking at alternative way for microinjection, including ours. Actually, one of the key players for the study is going to be your TA this afternoon, Andrew, who sits there. And so the idea is to see whether we can use electroporation to improve, to deliver Cas9 system into zygote. And so in the first Howie Wang's group actually look at the possibility to electroporate Cas9 mRNA and guide RNA. They reported limited success, but it's actually now so easy to make it work. And then this year, Howie's group and as well as my group actually published a method to use Cas9 RNP electroporation and then deliver CRISPR system into the mouse zygote. And this enable not only very efficient NHEJ and HDR editing, but also significantly improve the embryo survival. So I told you 10 to 20% survival birth rate is very common for injection procedure. But in the electroporation procedure, you can improve that by 2 to 3-fold. So the idea is that you pre-assemble Cas9 complex, so Cas9 protein and guide RNA. You pre-assemble the RNP complex. You pull many zygotes together, and then you just electroporate using a desirable scheme. And then after electroporation, you can directly transfer this into recipient mouse. And then this procedure enable an average lab like my lab to be able to perform CRISPR genome editing and greatly decrease our dependence on transgenic facility. For those of you who work with mice, you always have to wait for months before your construct can be injected, right? So I want to show you a video and just to hopefully convince you how easy this could be. And this is actually Andrew performing the experiment. So you actually put the embryo, you mouse pipette the embryo, you put them into a drop, and then you take RNP complex from another tube, just using regular pipette, and then you pipette in the RNP complex with your embryo drop, and you mix them a couple times, and then you put it into a cuvette, just like you electroporate bacteria. And then all you need to do is put it into an electroporator. We use a BioRigine pulsar electroporator, but you can use other more fancier electroporator, such as Lanza, for example. And then you just hit a button, and that's it. You don't need to sit there for hours and inject one by one. So I was very pleased with this development. So the results are amazing. So we take our proof of principle experiment, we take a guide RNA against tyrosinase gene, which is the most important enzyme for pigment synthesis. So if you biallelically inactivate tyrosinase gene, you will get albino mice. So this is our mRNA injection where, we get about 60% biallelic editing, but this is our CRISPR, we call CRISPR Easy, which stands for CRISPR RNP electroporation of zygotes. And then we got about 88% biallelic editing. And then we designed a pseudo HDR experiment as well. We achieve about 46% HDR efficiency. What I want to draw your attention is this bar. So under this particular condition, we have a lot of 88% biallelic editing, and we also have 2 mice that are mosaic. I only have 1 black mice, which are also edited. It's a heterozygous. So if you look at this, what's amazing about this technology is you get 100% delivery. So every single embryo got the CRISPR system through electroporation. And the other really amazing advantage of the system is if you look at the viability of the embryo. So shown here, the gray bar is we did a side-by-side comparison for tyrosine guide RNA injection. And then this is the efficiency, it's less than 10% for the pup survival. And then if you go look at the data across a number of the mRNA injection project in the Berkeley transgenic facility, you get between 10 to 20% embryo survival. But if you do CRISPR Easy, these 2 different conditions, you can significantly improve the embryo survival. So instead of collecting 200 embryos, just doing 1 CRISPR project, you can do multiple CRISPR projects. So it's fast, it's 100% delivery, it's efficient. I think it's slightly more efficient than injection. I wouldn't think that the efficiency of editing is a lot more. It is slightly more. But the most amazing advantage for this is about 3 times increase in embryo viability. This enables you to do more experiment. And it's easy, it's economic. Actually, this is a key reason why we developed the technology, because we were so broke at that time. And then it can be done in a high throughput manner. So you can actually pull, dozens of embryos together and electroporate in 1 shot. So the CRISPR Easy challenges. So I think the challenge is that you can really only reliably use this technology for relatively easy editing because it's very difficult to deliver large circularized plasmid donor by electroporation into zygote. We're hopefully working on this and try to find out strategy. And then it would be great to look into other Cas9 variants to see whether we can improve the system from the delivery standpoint, and then also try other mammals. So mouse embryo are among the easiest mammalian embryo to manipulate. It's very sturdy. You can manipulate them in many different ways, but we're not so lucky with other mammals such as cat or cow, for example. And so it would be great to this improve in survival, maybe really advantages in other mammalian species. Okay, so let's talk about genomic variants. So like I said, most of the genomic structural variants were done in ES cells. And this is a study that's reported last year in which they identify a 500 kb deletion region in a very rare patient in the introgenic region of this gene called LAP4. And this patient have malformation of the limb. So this is actually show a triangular tibia, which is very shortened compared to what you normally see. So originally people thought, oh, this is a loss of function mutation. Let's knock out this LAP4 gene in mice. Knock it out, absolutely no phenotype. So, and then using CRISPR, they can actually design a large 500 kb deletion initially by mouse ESL editing and then generating the mice. And then they found that this 500 kb deletion, which exactly mimic what happened in the human patient, gave rise to a similar phenotype. They see truncated femur and triangular shaped tibia shown over here. So, so this experiment really suggests that this deletion are most likely generating a kind of a gain of function effects for this gene, because this deletion gave rise to a truncated LAP4 protein whose phenotype is not easily read out by just straight knockout. And then also Matthew Meison's lab have who actually are working on mouse cancer genome have identified a lot of translocation events, large deletion events, inversion events in human cancer. So he really wanted to be able to use CRISPR to model these events. So what they did is they, in 293 cells, in proof of principle experiment, they were able to design 2 different guides on 2 different chromosomes and then trigger the same kind of chromosome translocation events you will see in human cancer. You could also do inversion. This is a very famous case in lung cancer, non-small cell lung cancer, in which the ALK kinase and EML4 genes are transcribed on different direction on the same chromosome. And in a subset of lung cancer patients, there's an inversion event happen, which enable the production of a fusion gene between EML4 and ALK. And then this fusion gene leads to aberrant activation of ALK. So this group of patients actually respond to ALK inhibitor, which is very effective in clinics. So Matthew Myerson was able to, in 293 cells, was able to recapitulate this inversion events. And then what's also very interesting is in another inversion events equally frequent in human cancer, this is a pericentric inversion. So they were also able to achieve this pericentric inversion that involve 2 different arms of the same chromosome. This is all in cell line, okay? So later on, a group in Sloan Kettering, Andrea Ventura's group, actually bring the system in vivo. So what they did is they want to mimic this chromosome translocation events for EML4 and ALK. And then what they did is they deliver adenovirus that encode both the guide RNA and the Cas9 by intratracheal injection. So the adenovirus only infect lung epithelial cells. And then by doing so, they were able to recover tumors that carry this reciprocal translocation events. And then these tumors express these fusion genes, and then they display the histopathological features of the corresponding human patients. So as you can imagine, if you do this using tissue cells, it's actually very difficult for you to genotype, right? How would you say, okay, you have all these events, and how can you really show that it's this particular reciprocal translocation events that really lead to your phenotype? So they're very lucky in the way that they are looking at cancer cells. The initial efficiency is extremely low, but because of the growth advantage of the reciprocal translocated cells, they were able to amplify the clone. So when they were also able to show by fish experiment that in the wild type situation, the 2 ALK probe are close together, but in the knockout situation, the 5' ALK probe and the EML4 probe are close together on the chromosome region. So that's a way that they can show. But this potential heterogeneity of editing events and off-targeting events could be difficult to interpret in this kind of tissue mouse modeling using CRISPR. Okay, so yeah, I pretty much covered this. And so I just want to say that in addition to lung, there are many other tissues that are susceptible for tissue engineering. So you can actually deliver plasmid DNA and virus by hydrodynamic injection. This is injecting a large volume very quickly through tail vein into the mice so that everything ends up in liver. For lung, you can do intratracheal injection, intranasal incubation. And then for hematopoietic system, you can do ex vivo because you can culture hematopoietic stem progenitor cells in culture, manipulate them, transfer them back into recipient mice. For brain, you can do sterile tactic delivery by AAV. And then recently, there are also development of the inducible CRISPR mice. How much time do I have, Dana?
5 minutes.
Okay. Yeah, so let me just give you some example. This is a study published by Tyler Jack's group in which they do hydrodynamic injection of Cas9 plasmid DNA that encode Cas9 and then also the guide RNA. This is actually two guide RNA that target two important tumor suppressors, P10 and P53. What I want to draw your attention is over here. So this is a control where you don't see tumor formation. This is the Cas9, the CRISPR editing, and then this is actually tissue-specific conditional knockout for P10 and P53. So you can see it pretty much recapitulate the oncogenic phenotype. This is a study done by Feng Zhang's group where they actually target brain. So they use two AAV vector, one encode Cas9 and one encode the guide RNA. And this is largely because Cas9 are too big. It's difficult to encode everything into one AAV vector. So they're targeting MECP2. So you can see in their targeted animal, you can see a great reduction of MECP2 by IF staining in the dentate gyrus of the hippocampus. So then the effort to make knock-in mice, there are two popular knock-in mice that people use. One is developed by Feng Zhang's group, in which they use ROSA26, which is a ubiquitous promoter. So you can actually use a ROSA26 locus. You can target a fragment that encode a strong CAG promoter driving the lock-stop LOX cassette, and followed by Cas9 and GIP. So this enable you to do inducible Cas9 expression. So if you have tissue-specific CRE, you can excise this lock-stop LOX cassette, and therefore Cas9 will be driven by the constitutive CAG promoter in these mouse. So this is targeted into the ROSA26 locus. So they actually use this model and then to target, I thought that was a very interesting experiment in which they have guide RNA that target both P53 and LKB1, and these are tumor suppressor. But it's also interesting that they use a guide RNA that target KRAS gene, and then they also encode a donor template, HDR template, that direct this guide RNA to generate a KRAS activating mutation. So when they deliver this intratracheally into this CRISPR mice, they were able to generate tumor formation. So that was, I would think that the editing efficiency is extremely low, but again, they're working on cancer model and that get amplified to become a homogeneous clone. Another interesting thing I want to say that you can see, they look at 4 weeks after the delivery of the guide RNA, as well as 9 weeks. You can clearly see this enrichment in 9 weeks in terms of the editing event. So it's really the formation of tumor that amplify their desirably targeted clone. Another model was developed by Scott Lowe's group. They made a TET-inducible Cas9 allele. So they actually use a collagen 1 locus, which is a locus that often have open chromosome. So what they did is they have this targeting vector that targets the guide RNA, as well as a TET-inducible promoter driving GIP Ares Cas9 or the NICase. And then using this model, you are able to induce Cas9 expression in a tissue-specific manner by the DOX treatment. So in the proof of principle experiment, again, they actually targeted the intestine. So they targeted both APC and P53. These are all very important tumor suppressor. So they targeted them by NHEJ editing and were able to see expansion hypoplasia of the intestine. So the series of experiments really suggests the power of CRISPR editing in the somatic editing. So this is my last slide. Sorry to bear with me just one more minute. Clearly there are a lot of advantages because of the simple design, the easy manipulation, the rapid and ability to do multiplex editing. But there's also challenges. We really need more reliable guide RNA design, and I think the variation of the success you see can be largely attributed to the different guide RNA that you design. And then we really still depend on ES cells for very complex genome editing. So with the improvement of the CRISPR genome editing, we hope to be able to do this in one step. And to achieve the the desirable editing. And then finally, the CRISPR editing can generate a variety of editing events that are just unclear to predict. So we also need a very precise way to identify the genotyping strategy, and then using mouse as a pre-implantation embryo mouse embryo as a way to screen for desirable editing events. We will be able to actually pre-screen the strategy, so we don't have to generate mice. We can just screen the editing events in the mouse embryo. So if we can improve on the precision genotyping strategy, we will have a much greater power with generating CRISPR editing in mice. For the somatic mouse models, we really need a better delivery system to target more tissues, more cell type, and also we particularly need a smaller Cas9 variants for viral delivery, and that seems to be a lot of challenges, the root of a lot of challenges in the somatic delivery. Off-target effects, and it's actually largely ignored up to now for somatic mouse modeling. Actually, we really don't understand the impact of the off-target events on this modeling. So I think more careful study are needed to characterize off-target events and how they impact those somatic genome editing, where you can now just cross out the off-target events by breeding. So also with the existing tool for CRISPR system for the mouse genetics community, we will be able to maybe combine CRISPR to generate more powerful technology to dissect biological question. So as a biologist, I really want to say technologies are a powerful tool for us to make discovery, but it would be really great that we can make real exciting discovery using this new technology rather than doing the proof principle experiment. I think the proof principle experiment are great so far, so I'm very excited to see what's going to happen in the next few years when people use this technology to make exciting discovery that they couldn't make in the previous years. And that's all. Thank you very much.